Hi, I'm Wendy Vandersteen, a research associate at the University of British Columbia. I'm currently involved with a project looking at assessing optimal rearing conditions for salmon within land-based recirculating aquaculture systems and trying to help facilitate the sustainable development of the land-based closed containment industry. Today, I'll spend the next 10 minutes or so just providing some information on the genetics of transgenic salmon, so how transgenic salmon are created, and some of the impacts that this transgene has on growth and other characteristics. So first, before I get into the details, I just want to acknowledge Bob Devlin, a research scientist at the Centre for Aquaculture and Environmental Research in West Vancouver. He's been the driving force behind the development of this transgenic strain and the development of a risk assessment program for the use of these fish in research and industry. Also, Fred Sundstrom and Marty Lomas have been quite involved with the ecological risk assessment. So there's an ever-increasing list of fish species that have had some type of transgene inserted into their genome. The purpose of this transgene can vary from things such as control of invasive species to developing strains that can be used for environmental monitoring. However, the most focus of this technology is to improve the growth rate of these fish for the aquaculture industry. So why is growth rate so important for aquaculture producers? Well, ultimately, if you can increase the growth of a fish, you can improve the production efficiency. So increased growth means reduced time to market. It can also improve the feed conversion efficiency, so you can get the same size of fish with less amount of feed. It can also tend to decrease or increase the survival and decrease husbandry costs associated with rearing these fish. If we consider growth of a fish, we can see that it's controlled by a large number of factors. Of course, you have the environmental factors, so things like rearing temperature that the fish is exposed to. Growth will also be heavily influenced by what the fish is eating, so the quality and the quantity of food. It's also strongly controlled by hormones fluctuating within the fish itself. One hormone in particular, somatotropin, or growth hormone, which I've indicated just with GH, has a very strong effect on growth. If you increase growth hormone, you can increase growth of the fish. Growth hormone is also involved with a lot of other physiological pathways in the fish, so it can affect not only growth, but many other characteristics. So very basically, in order to make a transgenic fish, you have to take a promoter, which regulates the expression of a gene, and you have to fuse that to your gene of interest, which in this case is the growth hormone gene. That produces your gene construct, or your transgene, which you then insert into the fish. And for this specific strain, you end up with unregulated expression of growth hormone. So I'll go through this in a bit more detail. This strain of fish incorporates parts of two different genes. So we have the metallothionein gene, which you don't have to worry about the name of the gene. It's just a protein that's expressed widely throughout the body, so in a number of different tissues. And it's expressed at a moderate level. The other gene is the growth hormone gene that we're interested in. Typically, it's expressed only in the pituitary gland, so a very specific location within the fish. And it's also regulated seasonally, so you might get increased production in the summer, decreased production in the winter. And it's also affected by other hormones in the body, and in particular, growth hormone itself self-regulates. So as growth hormone production increases, it'll feed back and stop any further production. Now, if we take the promoter, so the part that regulates that metallothionein gene, and connect it to the growth hormone gene, you now end up with growth hormone being produced throughout a number of tissues in the body, and it's now no longer subject to regulation from seasons or self-regulation from other hormones within the body. So you end up with unregulated production of growth hormone. Just to put this into context, if we consider the genome of the salmon, and if we write that genome out on 8.5 by 11 pieces of paper, line those pieces of paper up, it would stretch for about 330 kilometers, so approximately from Vancouver to Kamloops. Inserting that transgene into the fish is like taking one of those pieces of paper and swapping it with a different piece of paper. So it's a very small change, but this change can have very strong impacts on growth. So once you inject the transgene into the eggs, let the fish grow, you can see extremely rapid growth even after only 6 months. So the big fish on the top are the transgenic salmon, and the smaller fish at the bottom are non-transgenic, and these are coho salmon after 6 months of growth. You see similar effects with other species. This shows the impact of the transgene on rainbow trout. If you focus on the left-hand side of the figure, the large fish at the top indicate the transgene in wild trout, and at the bottom is the transgene, or the non-transgenic fish. So again, quite significant differences in growth. And this has been done for other species of fish besides Pacific salmon. It's found in Atlantic salmon, also Arctic char, catfish, tilapia, carp, mudloach. And in all of these cases, you see very rapid growth of the transgenic fish compared to the wild fish. Now, besides increased growth, you can also see quite significant differences in behavior. This figure shows a picture of coho salmon within a tank. On the left-hand side, you have the wild coho salmon. They're grouped very tightly together at the bottom of the tank. On the right-hand side are the transgenic salmon, and you can see they're much more widely dispersed in the tank. They're much higher, closer to the surface of the tank. They're ready to start feeding, and that's what they do. They're very highly motivated to eat. They're not as concerned about risk of predation as the wild fish. Aside from differences in growth and behavior, you end up with a whole suite of other physiological differences. You can have altered disease resistance, so sometimes the transgenic fish tend to have reduced disease resistance. You can have differences in swimming ability, altered feed conversion efficiency, which tends to also be associated with actual differences in their intestinal structure. You can also end up with differences in the muscle fiber and size, so the fillet itself can have some different structure to it. There have been some studies done on the proximate composition of the muscle of transgenic versus wild fish. So this is the makeup of the fillet. One difference of interest is in coho salmon, you can end up with either increased or decreased percent lipid in the muscle compared to non-transgenic fish. And this depends solely on how the fish was raised up to sampling. So whether it had a high food diet or low food diet will affect those differences. So since these fish are being created for the aquaculture industry, we have to ask whether we should be concerned about the use of these organisms for this industry. There's a long history telling us that many fish extinctions have occurred because of introductions of foreign species. So we know that major ecological issues can arise, and in this case, they're very difficult to predict. If these fish are being used in the aquaculture industry, there's always the potential risk that there could be an escape into the natural environment. And the big issue is that once this occurs, it's essentially impossible to get these fish back out of the environment. In order to do a proper risk assessment of what the potential impacts could be if these fish escape, the best way to do that is we need to know data on their fitness in the actual natural environment. But this is impossible. We can't do this. So one of the major things done at the Center for Aquaculture and Environmental Research, directed by Bob Devlin, is to try to develop ways to do a proper risk assessment without having to actually get the data from nature. One of the biggest parts of the program are trying to replicate natural environments within the closed containment culture environment. So trying to restructure tanks and build streams within the lab that we can use to recreate more natural rearing conditions for these fish. So we can try to see what the transgenic fish might look like and what their fitness might be if they were in the natural environment. One of the major findings from all of the research done over the years is that environment has a huge impact on the phenotype of these fish, so what these fish look like. It has a huge impact on their fitness. If you take a transgenic fish, rear it in a culture environment, give it lots of food, it'll grow very quickly. If you start to restrict the food that they get, or if you start to rear them under more natural conditions, they start to look more like fish from the wild. And the issue with this is that risk assessment has to be done on a case-by-case basis. So you can't come up with some general trends and assign it to all situations. It's very dependent on the environment. So in closing, I just want to acknowledge all the people who have participated in this research. And I'd like to thank the Vancouver Aquarium and Oceanwise program for giving me the opportunity to speak. Thank you.